The overall goal of this procedure is to examine bacterial uptake by M cells located within the intestinal mucosal immune system. The first step of this procedure is to expose and ligate the intestinal Peyer's patches of an anesthetized mouse. Fluorescent bacteria are then injected into the ligated area. An hour later, the Peyer's patches are excised. The cells in the tissue are then permeabilized and stained for the M cell marker GP2. Finally, the samples are observed using a confocal microscope, and bacteria being transcytosed by M cells can be visualized.
In addition to providing insight into the molecular basis of the bacterial transcytosis by M cells, this method can be applied to other systems. For example, this protocol could be used to assess the cell permeability in inflamed intestine using fluorescent microbes.
Using a sterile glass spreader, streak the fluorescent bacteria on an LB agar plate and incubate the plate at 37 degrees Celsius until single colonies are visible. Pick a single colony off the solid agar and inoculate it into 2 milliliters of fresh LB medium, then culture the bacteria on a shaker overnight at 37 degrees Celsius. Once the starter culture has grown, transfer 500 microliters of bacteria into 4.5 milliliters of LB medium and incubate this new culture at 37 degrees Celsius for 3 to 4 hours, or until its optical density at 600 nanometers reaches 1, indicating that the bacterial growth is in log.